Hi, this is Pete Yosey, founder and editor of Epigenie, and I'd like to welcome you to today's webinar titled Multiplex Profiling of Epigenetic Modifications, Measuring Assay-Specific and Off-Target Effects in the Same Sample. Today's webinar will be presented by Anne Jelinek, a senior research scientist from Active Motif. Dr. Jelinek's background includes over 15 years experience in development of reagents and assays in the epigenetic research arena at both Upstate Biotech and Active Motif. She's commercialized hundreds of polyclonal and monoclonal antibodies to key epigenetic targets, including histone post-translational modifications, and leveraging these antibodies for use in assays, including the first-to-market chromatin immunoprecipitation assay, histone acetyltransferases, and deacetylases enzymatic assays, as well as the Histone H3 PTM Multiplex assay for the simultaneous detection of multiple histone post-translational modifications. This webinar today is brought to us by Active Motif. Active Motif is committed to helping researchers study chromatin biology and epigenetics through optimized research systems, including chromatin IP kits and reagents, validated antibodies to histone modifications and chromatin proteins, kits for analyzing DNA methylation and DNA damage, as well as an ever-increasing range of epigenetics research services. So as always, we'll be having a question and answer session after the presentation. So throughout the presentation, if you have a question that pops in your mind, please put that into the question box, which should be in your panel, your GoToMeeting webinar panel, should be on your desktop, and I will pull those together and pose them to Dr. Jelinek at the end of the presentation. So on that note, I'll turn things over to you, Dr. Jelinek.
Thank you, Pete, for that introduction, and to you, the audience, for joining me today for this presentation. I will start with a brief overview of what I will be covering today. I'll start with the requisite introduction of epigenetic mechanisms and how they pertain to the regulation of gene expression. I'll then introduce the Histone PTM multiplex assay that we've developed and the Luminex XMAP technology on which it's based. Then I'll describe how one goes about setting up using the assay and show some data sets that demonstrate the assay specificity, sensitivity, and reproducibility. And then I'll close the presentation with an announcement of the availability of this particular assay as the newest service in our portfolio of epigenetic services. Epigenetics is broadly defined as changes in phenotype that are heritable and do not involve changes in the DNA sequence. Epigenetic mechanisms are involved in a variety of cellular processes that include DNA imprinting, repair, aging, and of course, gene expression. In this slide, we illustrate how the epigenetic mechanisms of DNA methylation, histone modifications, and non-coding RNAs work in concert to regulate gene expression. DNA methylation occurs at CPG-rich clusters or islands located at gene promoters, and when present, methylation is responsible for repression of gene transcription. Histone acetyl marks, chromatin modifiers, and non-coding RNAs work in concert in context-specific manners with DNA binding transcription factors, which bind DNA sequences in a context-specific manner to either allow for the transcription of a specific gene or repress that gene. As we all know, all gene expression occurs in the context of the nucleosomal structure of chromatin. A nucleosome is, of course, DNA wrapped around 8 copies of histone molecules, of which consists of 2 copies of histone H2A, H2B, H3, and H4. Histones are small basic proteins that have unstructured amino terminal tails that are extensively modified by post-translational modifications. The modifications include phosphorylation of serine, threonine, and tyrosine residues, methylation of arginines and lysines, acetylation of lysines, and ubiquitylation of lysines. There are additional modifications that aren't on this slide, but to include all of these would just take too long. Histone modifications are not static. There are some marks that change throughout the cell cycle, while others are more stable and heritable, spanning generations. There are three classes of proteins that interact with the histone code. There are the readers that recognize either a specific mark or specific combination of marks. Then there are those proteins which deposit the marks, which are colloquially known as the writers. And then those enzymes that remove the marks, and those are called erasers. The readers and the writers and erasers are listed in this slide. Those that are in green type are associated with activation of gene expression, whereas those that are red are associated with repression. Dysregulation of any one of these proteins results in, at the molecular level, altered patterns in gene expression, which at the cellular or organism level ultimately impact homeostasis and therefore play causative roles in either the establishment or progression of disease. This dysregulation is also manifested at the cell level as changes in the global levels of histone modifications. And these changes have been observed in a variety of different states that include cancer, aging, psychiatric and neurological disorders, autoimmune and inflammatory diseases, and following exposure to environmental stressors or toxins such as arsenic and cigarette smoke. Specifically in the context of cancer, decreases in dimethyllysine 4 of histone H3 correlates with high recurrence rates in prostate cancer and poorer survival rates for patients with lung, kidney, breast, or pancreatic cancer. The changes, though, are not always decreases. In the case of gastric adenocarcinomas, increases in trimethyllysine 9 of histone H3 have been shown to correlate with poorer survival rates. Now, the dysregulation of these marks also means that the global histone levels make for attractive readouts for drug screening applications as pharmaceutical strategies are developed to target these proteins that are dysregulated in this particular epigenetic mechanism. The most common methods used for monitoring changes in histone modification include ELISAs, which require microgram amounts of sample, are relatively high throughput, but are limited to the analysis of only one mark at a time. In contrast, immunohistochemistry or immunofluorescence, depending on the instrumentation that you have available, you can analyze multiple marks simultaneously. However, throughput is relatively low due to the laborious and technical nature associated with sample preparation. Western blots, while technically simple, do also require large amounts of material, are low throughput, and again are limited to the analysis of a single mark in a lane or an entire blot. Collectively, the challenges facing global mark detection methods are that they require large sample amounts, are low throughput, and are limited to the analysis of a single modification. Because of these challenges, the mining of the wealth of information that can be obtained from the histone code and its dysregulation has been essentially limited to merely scratching the surface. To address this challenge, we looked into other areas of biology to see what solutions have been come up with. Looking at cytokines, which are used as readouts for immune system function, we noticed that the standard one-target ELISA has been replaced with multiplex versions. We thought that the same approach could be leveraged towards the histone modifications by multiplexing histone ELISAs. And we decided to use the multiplexing platform that has been the most widely used in the cell signaling and cytokine fields, which was developed by Luminex. Luminex calls their multiplex technology the XMAP technology. It utilizes small polystyrene beads that are coated with reactive groups that allow for the addition of a probe or a biological reagent. These beads are also contain a mixture of two dyes, a red and an infrared dye. And all beads that belong to the same color ratio of dyes belong to a specific bead set, which is then identified by the software of the instrument with a unique bead number. We have developed a panel of histone H3 specific beads for using this platform, and they include a total histone H3 analyte for normalization between samples. We have specifications for methylation sites at either lysine 4, 9, or 27. Acetylation includes a panacetyl bead or one that is specific for lysine 9, 27, or 56. And we also have two phosphorylation marks for serine 10 and threonine 11. The multiplex assay is performed essentially in the same manner as a traditional ELISA. You start off using a 96-well plate. It requires roughly 3 hours to complete. Now, unlike a traditional ELISA, the assay allows for the comparison across multiple samples and multiple PTMs simultaneously. Magnetic beads enable the use of a plate magnet, which facilitates washes. And at the end of the experimental manipulations, the plate is then read in a Luminex instrument. Unlike the traditional methods for detecting histone modifications, this assay only requires nanogram amounts of sample and is compatible with purified core histones or cruder acid extracts from either fresh or frozen cells or tissues. When working with frozen cells, cells from 24-well plates or larger generate enough material for use in the assay, whereas with fresh cells, you can go straight from cell culture to assay in the 96-well plate format, making this platform extremely attractive to the high throughput and drug screening areas. To give you an idea of what is involved with regards to hands-on manipulations, to perform an assay, you start by creating a master mix of the beads that corresponds to the marks of interest. And you dispense the beads into the wells of an assay plate. Each bead is coated with a distinct modification state-specific antibody whose specificity in the context of this assay has been extensively validated during development. I won't touch on the nature of that testing now, but if anybody has any questions at the end of the seminar, I'd be happy to elaborate on that further during the Q&A. Histone-containing samples are then added to the wells and incubated for an hour. Excess unbound material is removed via a series of wash steps that are performed on the plate magnet. The plate magnet retains the magnetic beads, and so the beads are not lost during the wash steps. Next, a two-step detection system is used to determine how much histone has been retained on the beads. We first start off with a biotinylated reporter antibody specific for the carboxy terminus of histone H3, and that is then followed by the addition of a fluorescent reporter tag. The assay plate is then placed in the Luminex instrument, and the instrument reads two pieces of information from each bead. It first identifies which bead set that bead corresponds to, and then it quantitates the fluorescent reporter signal associated with that bead. A minimum of 50 beads from each bead set is analyzed in each well, and the median fluorescent intensity is then reported. Data is presented either as a PDF report or a CSV file that can then be exported into spreadsheets for analysis. In the next set of slides, I will first describe to you how you go about setting up using this assay, and then I'd like to illustrate with a few selected data sets the specificity, sensitivity, and reproducibility of the modification multiplex assay. The first question you need to ask yourself is, how much material do I need to use? It is important that you make sure that you are in the dynamic range for each of the histone modifications. Now, this has to be empirically established because histone modification levels do vary between cell types. And lysate requirements vary across histone marks. Some marks are more abundant, some are less, and that, of course, then will impact how much lysate you need to use in the assay. And then finally, there are going to be differences in sample preparation. Depending on what kind of extract you prepare, the percentage of histones that are present in the histone may vary. Here I show you data that's generated from acid extracts from 3 cell lines that were assayed with the entire panel of bead sets, and the data normalized for histone H3 levels. To keep the slide uncluttered, data from only 5 of the bead sets is shown. As you can see, the signals from trimethyllysine 4 and acetyllysine 27 are much slower than that observed with lysine 9-trimethyl or lysine 27-trimethyl beads. And this is in part due to differences in antibody avidity, but it is also due to the abundance of these marks in a cell. Lysine 9 and 27 are far more prevalent in a cell than 27 acetylation or lysine 4-trimethylation. To further illustrate that differences of these marks differ between cell lines, if you just look at trimethyllysine 27, you see that in MCF7 cells, there is less of that mark present relative to mouse 3T3 or human HeLa cells. When setting up an assay, we recommend that you start with a 2-fold dilution series of the lysate using the bead multiplex of interest as a means of gauging how much lysate to use. Here we see data for 4 of the bead sets from the same experiment as was shown in the previous slide to show how bead signal intensity varies with the lysate amount. As you can see for trimethyllysine 9 and pan-acetyl beads, that amounts over a microgram per well have saturated those beads, and you no longer see any responsiveness of those beads with increasing amounts of signal. The region of the curve that we call the dynamic range is where you see proportional increase of signal with increasing amounts of lysates. So specifically, if you are interested in measuring changes in trimethyllysine 9 or pan-acetyl on histone H3, you would want to use lysates in the range of 200 to 500 nanograms per well. By contrast, if you were interested in looking at trimethyllysine 4, you would need to increase the input lysate amount. In addition to the antibody specificity testing performed during the development phase of the assay, we've also performed a series of inhibitor studies as a way of demonstrating in-assay specificity of these reagents. Here we show data from HeLa cells treated with the class 1 HDAC inhibitor valproic acid. The acid extracts were tested with a 6-speed multiplex, and the raw median fluorescent intensity is shown. Across the samples, you will note differences in several of the modifications, but you'll also note that there are differences in the amount of total histone H3 present in the wells. Following normalization of the data to account for the total histone H3 differences, the changes in the histone modification levels become more striking. Specifically, we see the expected effects of increases in acetylation at both the pan-acetyl and lysine 9 sites. But we are also able to observe off-target effects. We are able to detect increases in lysine 4-trimethylation, which has in fact been reported recently in the literature as a pretty common effect of HDAC inhibitors. We also are able to see decreases in lysine 27 dimethylation, which was a bit of a surprise to us. In contrast to the amount of material that's needed in the Western blot that's shown here on the right panel, the 6-bead multiplex was performed with 125 nanograms per well. Here on the right, the same lysates were subjected to Western blot analysis as an independent verification of the multiplex assay. The top panel shows roughly equivalent loading of histone H3 using an anti-H3 antibody, and the bottom panel shows a dose-dependent increase in K9 acetylation levels with an acetyl-K9 specific antibody. So it's a more than tenfold savings in material by using the multiplex assay. Here is a second inhibitor study to highlight the specificity of our lysine 9 and lysine 27 trimethyl beads. In this case, HeLa cells were treated for 11 days with low doses of the EZH2 inhibitor GSK126. EZH2 is the methyltransferase that deposits methyl marks on lysine 27 of histone H3. Again, on the left are the multiplex data, and the right the Western blot. And you can see that the data mirror each other very nicely. You see a decrease in lysine 27 trimethylation in the Western blot, and a similar drop-off in signal in the multiplex assay, but no change at lysine 9. And again, 800 nanograms of material is needed to generate this data. This technology can also be used for IC50 determination, again using HeLa cells and an HDAC inhibitor, this time SAHA. We see an IC50 for both the pan-acetyl and K9 acetyl beads that closely corresponds to what is reported in the literature for this inhibitor in this particular cell line. To further illustrate the compatibility of this assay in high throughput applications, I've included this particular data set. Here, HeLa cells were seeded at 1,000 cells per well in a 96-well plate and cultured for 24 hours prior to the addition of valproic acid, which was left on the cells for an additional 24 hours. The cells were then lysed in the cell culture wells, and 1/10 of the lysate was transferred directly to the assay wells for the multiplex analysis. We see the expected increase in signal of acetylation with the pan-acetyl bead, and we see that same modest decrease in lysine 27 dimethylation as we showed in the previous data set. Finally, in this last data slide, I show you the reproducibility of this assay. The HeLa cells that had been treated with coulsomid, a drug which arrests cells in mitosis, was repeated 3 independent times. And the data shown here on the left is the average of those 3 repeats, with the error bars being standard deviation. You can see the increases in serine 10 phosphorylation in the coulsomid-arrested cells, and you see the similar increase in the Western blot on the right. The histone multiplex assay was introduced as a kit for people to purchase and use in their own labs just a little over a year ago, and it has been very well received. We noticed that people started coming to us requesting to see if we could perform the assay for them. As a result, we have been working over the last few months to address these requests, and today we are pleased to announce that the histone multiplex assay is the newest addition to our portfolio of epigenetic services. 2 service options are available. The first is the traditional multiplex assay, for which data I have shown you today, that allows for relative quantification of PTM levels. And we will also be providing modification-specific quantitation using some of the recombinant histones that have a particular modification on them as the quantitation standard. Basically, if you send us your frozen cell pellets, we will generate the acid extracts, determine the protein concentration, and perform the assay or assays depending on the nature of the specific request and the targets that are analyzed, and then provide you both with raw data and analysis of the data. The amount of material that we will need from you will be roughly 200,000 cells per cell pellet, and we will also require some information on cell viability that will help us gauge the volumes in which to generate the acid extract, and so that all of the samples are at high enough concentrations for use within the Luminex assay. To summarize, detection of global changes in histone modification levels is a rapid readout of the dysregulation of epigenetic mechanism that centers around the histone code. It is manifested in numerous disease states, which makes it an extremely attractive readout in the area of drug development. The Histone PTM multiplex assay that we have developed allows for the simultaneous interrogation of multiple modifications. It is rapid and sensitive and compatible with high throughput screening of numerous samples. At this point, I would like to mention related kits available here at Active Motif. We have, of course, the Histone PTM multiplex assay that I've talked about today, but we also have developed transcription factor multiplex assays that measure the functional binding of the AP1 and NF-kappa B family members. We also offer histone purification kits that generate purified nucleosomal core histones. These are available in 3 different sizes that allow you to start, that depend on how much material you're starting with, whether it's a standard mini or microplate kit. And we also have the protein quantification kit that we use here internally for the quantification of protein concentrations in the multiplex assay. For further information on the assay or the service, please visit our website. At this point, I'd like to acknowledge the Active Motif scientists who have been instrumental in developing the assay and developing the assay as a service. I'd like to thank Epigenie for hosting this webinar, and I'd welcome the opportunity to answer your questions. Thank you.
Okay, thank you so much, Dr. Jelinek. That was a great presentation. And just to the rest of the audience, I have a few questions already here, but please go ahead if you want your question answered. Please go ahead and type that into your GoToWebinar panel. Again, thanks again, Dr. Jelinek. We just have a few questions for you.
Okay.
First of all, you mentioned that there's a kit that's been available for some time now in the service. Are there any differences between what a researcher would expect from the kit, other than the fact that they're doing it instead of you, between the assay kit and the service itself?
Yes. So one of the biggest differences is the fact that for the service, you would need to be sending us 200,000 cells as a frozen cell pellet. If you were to do it yourself, where you have the ability to then reduce the cell number requirements that you need to perform the assay, down to 1 to 2,000 cells in a 96-well culture plate. That is something that we just are not able to perform as a service, because those lysates are then too dilute, and the samples end up getting lost, stuck to the wells of the assay plate, and it's just not feasible to go down to that level when working with frozen material.
Gotcha. Okay. Good to know. And then also, can the assay detect more than one modification on the same histone?
At present, the assay does not contain any antibodies that are capable, that are specific for any bivalent modifications. And this is not that we haven't tried, but none of the antibodies that are available, whether they are ours or from other sources, have high enough avidity to be able to detect those marks. Then there's the other limitation, that the instrument will only read one bead at a time. Any beads that aggregate or form clusters of 2 or more are automatically discarded and not counted as readable data. So one PTM at a time per bead only.
Okay, good. And then can non-histone targets be co-analyzed in the multiplex assay?
That is something that we have not yet explored. One of the requirements of this assay is the use of acid extracts. So if the protein of interest is not present in that acid extract, then it will not be feasible to look at those additional targets.
Okay, gotcha. And then you alluded to the antibodies used in the capture and the other elements of the assay. How are those antibodies validated for you in the production of this product?
Okay, so when we develop a bead target, we go through a multi-step process to screen. We start off with a panel of as many antibodies for that same specificity to start with. And we start off by screening those antibodies on these Luminex beads that have either been coated with the recombinant histones that we sell that have the specific modification, or peptides that have those modifications. So we do a preliminary screen for specificity and select out, immediately eliminate those that have off-target specificity. We then take the top 3 or 4 leading candidates, conjugate those to the beads, and then we again perform rigorous analysis with either our recombinant histones that we have or with lysates. So we either do these drug studies where we can look at modulations that have been reported for that particular mark. We'll also do these peptide inhibition studies where we'll take a whole series of peptides, each bearing a single modification, and see whether or not the addition of those peptides will compete off or eliminate the signals that are associated with those beads.
Okay, interesting. Pretty thorough then.
Very.
All right. And then any plans to expand this into other core histone targets outside the H3 space?
Yes, we do have additional targets in development still in our R&D group. We have more targets for histone H3, specifically lysine 36 trimethylation will be available shortly, and we are developing content for histone H4, but those are still in the R&D phases, and we hope to be able to generate those in the coming months, both as part of the kit offering and as well as include them in the service.
Great. And I may have missed this, but what's the typical turnaround time on the service for the assay? So from receipt of sample to data.
Well, the turnaround time is something that we are in the process of still establishing. I imagine that we will first have detailed discussions with the customers as to what the nature of the project involves and kind of information they're looking for. But we imagine 2 or 3 week turnaround time is something that we'll be able to meet for in certain circumstances.
Gotcha. Okay. Well, it looks like you're off the hook now, Dr. Jelinek. Thanks again for a great presentation, a great discussion, and thanks again to our sponsor, Active Motif. And finally, thanks to all of you in the audience for taking time to listen in. We all know you're busy, and so we appreciate you taking some time out and hearing Dr. Jelinek's great informative talk here. If you've enjoyed this seminar, you'd like to see a video recording of the session, it'll be published within the next few days in the webinar section of our site. And there you can also go and view other webinars and past other technical presentations that we've had. So again, thanks again, Dr. Jelinek. We appreciate your time, and to the rest of you, until next time, good luck in your research, and goodbye from here at Epigene.
Thank you, Pete. Bye-bye.